Okay, so in this lecture, we're going to do a rundown of Chapter 6, which is Life's Mainspring and Introduction to Energy. So we're going to be talking about a lot about energy in this lecture, and I'm going to define what energy means in just a second. By the way, sorry if it's a little noisy. I'm in my lab right now. Well, it's not my lab. It's Dr. Ryan Hill's lab, but it's the lab where I work. And the refrigerator is pretty noisy. Our -80 needs to be thrown away. So I apologize if it's loud. So energy is central to life. And what is energy? And your book gives this analogy of the music box. And the music box analogy is that if you crank up a music box, it's going to start producing sounds because you have put energy into it. And eventually, that energy is going to run out and the music will stop. So in our case, the hand on the crank that has gotten life going is the sun. So a huge majority of our energy comes from energy from the sun. So the solar radiation travels through space, gets to our planet, and provides us with energy. And we'll talk about kind of why that's an important idea in just a second here. But part of that process is that energy tends to run out. It tends to run downhill. And as we're going to see in just a second, all energy in the universe tends toward what's called entropy. And we're going to discuss that here very shortly. So before we can get into that, we kind of need to talk about, well, what is energy? How do you define energy? And I think we all have kind of an intuitive sense of what energy means. But energy in biology is, we can think of it as this capacity to do work or this capacity to create motion against an opposing force. That is how we're defining energy. So energy is the capacity to do work, which can be thought of as moving against an opposing force. There are two main forms that we're going to talk about: potential and kinetic energy. Potential energy is stored energy, and in biology, a lot of that energy is going to be chemical. So if you are to take let's say you take a pencil and you hold it up in the air, that pencil has a lot of potential energy. You lifted it up there because of gravity. Now, if you want to convert that potential energy to kinetic energy, all you have to do is let the pencil go and it will fall to the floor. And thus you have converted potential to kinetic energy. So like it says here, it's like a rock on a hill. If you give it a little tiny push, it's going to roll down the hill. Or it's like a lump of coal. And probably most of you have not burned coal ever. I never burned coal other than like charcoal. And what's happening with that coal is it has a bunch of energy stored in its chemical bonds. And that energy can be released by setting it on fire, and it will burn up and release all of that energy. Okay, so that's potential energy. It's stored energy, energy that can be used. We also have kinetic energy, which is energy in motion, which is like that rock rolling down a hill or the heat coming off of burning that lump of coal. It's you take potential energy and you tend to convert it into kinetic energy. So this idea of the first law of thermodynamics is that energy is neither lost nor gained. And this is true anywhere in the universe. Energy does not go anywhere ever. Well, it goes somewhere, but it is not created nor is it destroyed ever anywhere in the universe. So all of the energy that we started with in the universe is still here. None of it has been created. None of it has been destroyed. But it certainly can change forms. So that would be like taking solar energy, taking that solar energy and using it to make carbohydrates, which is what plants do all the time. So plants take solar energy, they suck it up with their pigments, they absorb it, and they convert it into potential energy. You can also think of it like a steam engine burning coal to power a train or to power, whatever device. Another example of converting energy is taking your car and turning it on, and you're converting that gasoline into motion. You're converting gasoline, chemical energy, into your car moving forward. And this notion of like going back to the first law of thermodynamics, this notion of energy is neither created nor destroyed might be slightly confusing. You might think, well, energy is lost all the time. It kind of must be. What about when I hit the brakes on my car, right? You're taking kinetic energy and you're just slowing down. You're not converting it, right? But you are, because energy is lost as heat. Whenever you convert energy from one area to another, whenever you convert it from potential to kinetic, or the other way, some energy is always lost to heat in those systems. And that brings us to the second law of thermodynamics, which is what happens when energy is transferred. So if you start with a piece of coal and you light it on fire, the reaction is going to end up with ashes and hot air, which is the kinetic energy. And basically, things are going from this ordered chemical bonds, these very ordered systems, and they're tending toward chaos, tending toward entropy, tending toward disorder. And this is true of all systems in the universe. All systems in the universe are continuously leading, or not leading, but they are continuously tending toward entropy. So you could kind of, at least for our purposes, you can think of entropy as just being heat. Okay, so basically what the first law is saying is that energy is neither created nor destroyed. It is only transferred, say, from potential to kinetic. And the second law is saying that all systems tend toward entropy, or all systems tend toward disorder. So, and you can think of this in terms of endergonic and exergonic reactions, which we're going to talk about in here in a minute. But coal does not spontaneously just reform from ashes and hot air, right? That would be the opposite of entropy. That would be going from disorder toward order, and you don't typically see that. So energy transformation only happens spontaneously when it goes from greater order to lesser order. So it is theoretically possible to take those ashes and hot air and convert them back to a piece of coal, but it would take a huge amount of energy. So that's not just going to happen sort of spontaneously. Okay, so the second law of thermodynamics describes how energy is transferred and states that when energy is transferred, it always results in greater disorder in the universe. So energy, or entropy, is a measure of disorder in a system. A lot of folks like to think of this as heat, or some people think of it as a available motion. I kind of think of it as just being disorder. So systems always tend toward disorder. So here is a little picture of transformation of energy. You are taking this chemical energy here that's stored in the form of coal, taking this chemical energy, and you are breaking those chemical bonds and using them to create heat, which is transferred into this water. This water starts boiling, and then we use that to spin a piston, or I'm sorry, piston-driven flywheel. It's going to spin this out, and we're going to use that to create some other kind of energy, probably electricity, but you maybe are using it to create motion or whatever it is. And at every single step here, energy is being lost to heat. So coal, you're lighting coal on fire, heat is lost. The transfer of heat from this coal to the water is not perfect, and so some of it's coming out of the side of your vessel. Then you have that steam, and that steam is right here in the vessel, and it's going to try to spin this piston-driven flywheel. Heat is lost again. So every time you change energy from one form to another, you always lose heat, and your entropy of your system is always increasing. So here are some examples of the energy efficiency of different systems. So a conventional power plant is 34% efficient, meaning that you only get 34% of the power transferred. So let's say this is, it says conventional, it doesn't specify, but let's say this is like a gas power plant. So only 34% of the energy stored inside of those chemical bonds is actually being used to make electricity. No system is 100% efficient, and that's definitely especially true when you're talking about energy. We also have human respiration of glucose, and it's 37% efficient, which is pretty efficient, actually. So 30%, 7% of the energy in the glucose that you're eating is getting converted to ATP, which we're going to talk about later. 37% is 37% efficient. So 7% of that chemical energy is getting converted to ATP. And an automobile engine down here, 25% efficient. Not too great, but not the worst. So 25% of the energy stored in your gasoline is getting converted into motion. The other stuff is, being lost to heat or being lost to, like when you hit the brakes, some of that energy gets converted into sound, actually. This is just talking about energy efficiencies of a few different systems. So some folks say, like, well, you have this second law of thermodynamics, which says that systems always tend toward entropy. And that is definitely true, which is kind of what makes this chapter a little bit sad, I think. And the reason it's sad is because in order for biological things to happen, we have to have energy from somewhere. And like I said at the beginning, our energy is coming from the sun. And one day our sun is going to, in fact, run out of energy. It's going to run out of the chemicals that are undergoing those nuclear reactions, and the sun will burn out, and life in our solar system will cease to be. Now, that's not going to be anytime soon, but it is in fact inevitable. So biological activities do in fact generate entropy. We generate heat when we try to do stuff. A high order of organisms decreases energy, which requires this huge energy input. And like I was saying, the reason we're able to do that is because of the sun. Because the sun is providing us all of this energy, we are able to sort of we're not defying the second law, but we have energy input, which is allowing us to build things and do things. But again, that is temporary. Okay? That is definitely temporary. And you should also realize that living systems do in fact contribute to this increase in entropy. We do contribute to this increase in disorder of the universe. So we are well within the confines of the second law of thermodynamics. This is just some more examples about energy reactions and cycles, talking about how we do in fact create more entropy. So when we do things like synthesis work, making carbohydrates, we are creating more ordered, more complex things than when we started with, because carbon dioxide gas, which is where most of our carbohydrates come from, all of our carbohydrates come from, it has way more entropy than a very, very ordered carbohydrate. So carbon dioxide, way more entropy than carbohydrates. And so we have to push that reaction uphill. And I'm going to show you a couple of energy diagrams so we can talk about what it means to push uphill. And it says they fight the second law of thermodynamics, but they really don't. They're totally in line with it. We are just getting provided a huge amount of energy from the sun. We also break down carbohydrates into carbon dioxide again through the process of cellular respiration. Okay, and that is what is called an exergonic reaction. It increases entropy. Exergonic, organic, you can think of this as exit. Energy is going out. And these systems up here, this is called endergonic because energy is going in. And we'll talk about that in just a minute here. So on the x-axis, you have you can think of this as time as the reaction is proceeding. On the y-axis, you just have energy. It says Gibbs free energy. Don't worry about that right now. But this is an exergonic reaction because your reactants over here contain more energy than your products. So you could think of this as here's a lump of coal. Here's a lump of coal that has a certain amount of chemical energy, and then you light it on fire. You need to put some energy into the system. You need to get the rock rolling. You light the coal on fire, and it's going to produce some energy for you, and then ultimately at the end, you're going to wind up with ashes, okay? And so the products are far lower energy than the reactants. Those lumps of ashes, way less energy than that very ordered lump of coal, way less chemical energy than that very ordered lump of coal. In an endergonic reaction, the reactants have much less energy than the products. You can see that. So reactants are way down here, products are way up here. And so this is like taking a piece of maybe carbon dioxide and converting it into a carbohydrate, a higher energy molecule. And you can see there's a bunch of energy required to do this. You have to push them all the way up this hill. And these are actually defined using this concept called Gibbs free energy. I'm not as worried about that. I just want you to know that exergonic reactions, you can see there is an energy loss between reactants and products here. Whereas in endergonic reaction, there is an energy gain between reactants and products here. This one you have to put energy into. This one you have to put energy into as well, but ultimately energy is going to come out. So very often in life we couple our reactions, we couple endergonic and exergonic reactions, and the example it gives here is like the sodium-potassium pump, because ions must be pushed uphill against their concentration gradient. But at the end, you have ATP, so you are getting energy out, ultimately. Okay, so we often couple endergonic and exergonic reactions in life. So here is a pretty good, this shows this glycogen molecule. So glycogen is just it's a carbohydrate. It is a carbohydrate polymer. It's got a bunch of glucose molecules all bonded together. And when you break these glucose molecules up, energy comes out. It's exergonic. Whenever you break a bond, energy comes out. So breaking these bonds, you get energy out of this. And you wind up with glucose, because these are each individual glucose monomers. And you're going to break them up, and you will get energy out at the end of the day. If you want to reform these glucose molecules, if you want to put them back into glycogen, you have to put energy in. It takes energy to reform those bonds. And so you have to put energy in to form bonds. That is an endergonic reaction. Energy is coming in. So in life, the main energy source that we use is called ATP. And ATP is this really interesting chemical where you have these negatively charged phosphates, and normally if you put two negative things together, they're going to repel one another. But we've linked them together. We have linked them together, making this high energy compound. So it's kind of like, they use this jack-in-the-box analogy in the book. If you guys know what a jack-in-the-box is, you push down a spring, and there's a little person thing on it, and once you are ready, you hit a button or you crank a wheel, and that jack pops out. So you're forcing that spring down. You're forcing that spring down and generating a bunch of potential energy. ATP kind of works in the same way. You have a bunch of negatively charged particles that normally wouldn't be together. They're trying to get apart, but you're chaining them together until you are ready to use them. So you have three, it's ATP, triphosphate, and you have three of these stuck together. When one of them decouples, it releases energy. Okay, so you break the bond, like I was saying earlier, whenever you break a bond, it releases energy. And it forms ADP, and your body is able to use the energy from that phosphate breakage so that you can do life stuff. All right? So here's a picture of our ATP. This molecule here is called adenosine. This maybe looks a little bit familiar to you from the DNA section. But on your adenosine here, you have these three phosphate groups. You have these three phosphate groups. And if you want to do something, if you want to utilize the energy, you're going to break this bond right here. And when you break that bond, energy is going to be released. So it's important to fundamentally understand that whenever you break a bond, energy is released. And that is kind of one of the points of ATP. It has these high energy bonds, and if you can break them, you can get some energy out of it that you can use to do cell stuff or life stuff. But you can also reattach this phosphate by adding some energy. By putting some energy in, you can reattach this phosphate and get this high energy ATP molecule back. So again, exergonic, you're breaking bonds, energy is coming out. Over here you have endergonic, you're forming bonds, energy is going in. So ATP is used to perform cellular work, like transporting things or moving things around. For instance, if you wanted to move calcium up their concentration gradient into a muscle cell, you would need to use ATP. So again, if you remember from the last section, things tend to move from regions of high concentration to low concentration. That's that idea of diffusion. Here, if you want to take something that is in low concentration and move it into an area of high concentration, you have to put energy in. You have to. And the way you're going to do that is using ATP. So ATP splits into ADP and a phosphate, which is that downhill reaction, which is going to cause energy to be released. That energy is used to transfer phosphate onto a protein, causing a change in shape that is allowing calcium to cross the membrane. And if that was all very confusing, I have a picture that hopefully will clear some things up. So here's the outside of your cell out here. Here is the inside of your cell here. And your cell is trying to pump out these calcium ions. So here we have our transmembrane protein. It has its calcium binding sites. It has these locations where calcium just fits in. So the calcium is going to come in here and bind. And now it's just kind of stuck there for the time being. Comes in and binds to our enzyme. And then you're going to have ATP come by, and it's going to release one of its phosphates. There's a bunch of energy coming out here when this phosphate attaches, and that's going to cause this enzyme to change shape. So calcium ions came in, bound here. They're stuck there for the time being. Then you have ATP come by, interact with this protein, and cause those calcium ions to be released. And then ultimately the phosphate's going to, unattach, and the shape change is going to be released. The phosphate is released, the enzyme changes shape, and then it's ready to accept more calcium ions. So ATP, can be regenerated. ATP is used as energy currency for many reactions and is generated by other reactions. So inside your body, ATP, how is it regenerated? Well, you have to have phosphate and you have to put energy into it. The reason we like to use ATP rather than just heat is because we can store it for long periods of time. And then you just saw how ATP is used as this energy currency for different reactions. Like you just saw that enzymatic reaction where it's releasing that phosphate. Okay. We also produce ATP in our bodies through the process of cellular respiration, which we're going to learn about later on. So enzymes, as we hopefully learned in a previous section, a vast majority of enzymes are proteins. Okay. So in order for an exergonic reaction to happen, you need to put in energy, sometimes a lot of energy. And what enzymes do is they lower that activation energy. They lower the amount of energy required to get that rock rolling. So sometimes you need a whole bunch of different enzymes to do something. Sometimes you only need one. But these are all coupled into metabolic pathways. Okay. And a substrate here during this discussion is the substance that is being worked on by each specific enzyme. So here's a picture. Here are your substrates. You have this little yellow thing and this little red thing. They're going to go into enzyme A, and they're going to come out. They start as products. They're going to come out as this reactant, this weird-looking yellow thing with a little red nub on it. Okay, so that is enzyme A has caused this reaction to happen more quickly. Now, it's important to understand that eventually, if you gave them enough time, these two products would come together to form this. But that might take a really, really, really long time. So the enzyme just helps out by lowering the activation energy. The next thing that happens is this yellow spaceship-looking thing goes and interacts with enzyme B, and it converts it into something else. Converts it into this third thing here. And this third thing goes and interacts with enzyme C and converts it into the final product. So this is this metabolic pathway by which you've taken a yellow and a red thing and turned them into a slightly longer, curvy yellow and red thing. And this might seem kind of silly, like who cares? Why do you want to make a red or yellow thing? But this product could be an important another perhaps enzyme, or maybe it is some product that your other cells are going to need in order to help them survive. Okay. So, like I was saying, enzymes are lowering the amount of energy required to get that rock moving. So here is perhaps a reaction. You got to get the rock all the way to the top of the hill before you can let it go and have it slide down its energy gradient. So we're converting lactose to glucose and galactose here. This is what it looks like when your stomach processes milk. And this is going to happen over time eventually, but if you don't have the enzyme, it's going to take a long time. It's going to take a really long time. If you do have the enzyme, you see here it's binding to the lactose, it's binding to the products, and ultimately forming these reactants. And the energy is is greatly reduced when you have the enzyme. That's the whole point of enzymes. They make things easier by lowering energy. So enzymes also are catalysts, which means that they are not absorbed in the process of this reaction. So if you start out a particular chemical reaction, you put an enzyme in, theoretically you should be able to get back out the same enzyme at the very end. So like I was saying, almost all enzymes are proteins, and we talked about the different levels of structure of a protein. I'm not super concerned with you guys knowing the specific details of protein structure. I would just like you to understand that shape is going to be super important when talking about enzymes in particular. So here's a glucose molecule that is binding to this hexokinase enzyme, and you can see this is a digital representation of what the enzyme, what this protein looks like. And the glucose has a very specific site at which it binds. It's a very specific site at which it binds here. That site is called the active site. And sometimes you need something called a coenzyme. It's just something else that is helping the reaction to take place, or perhaps it's helping the substrate to bind to this protein itself. So here's an example. This is chymotrypsin. Chymotrypsin is a protease. Chymotrypsin helps you to break down proteins. If you're thinking about these are going to be stuff that's happening at like sort of the interaction of your stomach and your intestines. Okay, it's helping you break these proteins down. And what's happening here is you have this active site where the protein is binding, and it's only going to bind to certain amino acids, and it's bending it. It's actually bending this amino acid chain. It's bending this other protein and allowing water to be able to get in there and interact. It is lowering the amount of energy required by your body to actually break these proteins down. So I've been kind of harping on this a little bit, but your body will ultimately break these things down given enough time, but it could take, it could take years to just have the right conditions to get the right amount of energy involved for this to happen. So this enzyme is helping this reaction take place by bending that polypeptide, that protein, that amino acid chain, whatever you want to call it, allowing water to get in and allowing it to be broken up. So what factors influence the amount of product? So that is the amount of substrate available. Remember, the substrate is the thing that the enzyme is working on. So how much you start with is going to determine how much you are ending with. Molecules similar to the substrate can also bind and block an active site of a particular enzyme. An example is the nerve gas sarin, which was used in a germ warfare attack in a Japanese subway. It binds to this particular enzyme and breaks it down so that the particular neurotransmitter can't be recycled, and basically it screws up your nerves. Okay, so what's happening here, this is called competitive inhibition, and it's not complicated. It's just something that looks like the substrate, comes in and binds to the active site and gets in the way of it. So that's one kind of regulation of enzymes, is competitive inhibition, or it's just competitive inhibition. You also have allosteric inhibition or non-competitive inhibition. And what's happening with allosteric regulation or allosteric inhibition is that something is binding to your enzyme and it's causing it to change its shape, which is either changing the shape of the active site or it's changing the shape of the enzyme itself such that nothing can get access to it. So here you have this thing coming in. This is your substrate, and it's getting converted into your product. Okay, so here you have allosteric inhibition, where you have these products coming in, and they're actually binding to this. It's called an allosteric site, and they're making it so that these things can't bind to the enzyme anymore. And so if you have too much of these products, this enzyme will stop working. This enzyme will stop converting substrate to products. This is called a feedback loop, because if you're making too much products, your enzyme is ultimately going to bind to this allosteric site and stop. So again, all that's happening here is this substrate cannot bind in here because the enzyme has changed shape.